Okay, let's get started. 2 weeks. Let me give you a little preview of what's going to happen in the next 2 weeks. We have our fourth exam the Thursday of, not next week, but the following week, the last week of class. And that'll be the fourth lecture exam, okay? And for tomorrow, I'll give you a list of chapters and things like that that'll be on that exam. The final will be 2 parts. The final will be a lab exam, a second lab exam, and that will cover the Pea Glow lab and the Grizzly Farm lab. That's worth 40 points, okay? There will be 10 multiple choice questions on the final exam. Those are the same 10 questions from cell biology that you took at the beginning of last semester when you did Bio 134. I don't know if you remember, but you took a pre-test when you came in. And there were 10 questions from cell, 10 questions from botany, 10 questions from zoology. We're giving you those same 10 questions again at the end of the class to see how you do. So those are 10 multiple choice questions, 2 points apiece. So that's 20 points. So the final exam is only worth 60 points. What we've tried to do is try to a little bit take the pressure off the final exam, because it's very common for you to have all kinds of exams the last week of class, and then you turn around 2 days later and then you have all these finals, okay? So our big exam will be the fourth exam. And then the lab exam plus those 10 multiple choice questions, which you really can't study for. I mean, this is stuff you should know now after taking this class, okay? So preparation for the lab exam will be important. The last day of class, I won't be here, but Dr. Morton will be here, and he'll do a final review for the lab exam on Friday, the last day of class. Okay? So with that in mind, it is important that you kind of wrap your head around what we've been doing in lab with this Grizzly Pharmaceutical Lab. So let's step back for a second and take kind of a big picture look again, because I think sometimes you get so caught up in the details of what you're doing that you lose track of kind of what the big picture is and what this project is all about. So in the scenario, you have a job. What makes or breaks your job? What do you have to be able to do at the end of your project? What determines whether you still have your job or not? You've got to clone that H gene. Okay? That's your job. Why do you need to clone that H gene? Because we need a whole bunch of copies of that gene for other groups to use to study that gene and its products as a possible cure for Hanover virus. Okay? So your job is to provide them the stuff that they need for their research. So what does it mean to clone the gene? What do I mean when we say clone the gene?
Have the gene induced into a bacteria, and then that bacteria replicate many things.
Good. So basically cloning means copying. It's making many, many, many copies. What copies it for you? A bacteria copies it for you. Okay? That involves getting that gene into the bacteria. So every time the bacteria, in preparation for cell division, duplicates its chromosome, it duplicates this introduced gene as well. So you just stick the gene in there by itself? No, we need some kind of way to introduce it into the cell. We need a vector. We need some kind of transfer agent. And what is our vector? That plasmid. And what do we call the plasmid? Puck18. Okay? Why is Puck18 a good vector? Well, number one, it's pretty small. But number two, it's got 2 really important genes on it that are going to be useful in this particular process of gene cloning. What are the 2 genes? Ampicillin resistance, that's the BLA gene, the beta-lactamase gene. And what's the other gene? Some people mumbling it, no one wants to say. The LAC-Z gene. What does the LAC-Z gene code for? The enzyme beta-galactosidase. And what does beta-galactosidase do? One of the things it does is it hydrolyzes lactose, but it also hydrolyzes this substance called X-gal. What color is X-gal normally? It's clear or colorless. And when it's hydrolyzed, what color is it? Blue. Okay? So the combination of the ampicillin resistance gene on the plasmid and the the LAC-Z gene acting on the X-gal in the environment gives us a way to select for whether there was successful transformation or not, and whether the transformation occurred as a result of a recombinant plasmid. What's the difference between a recombinant plasmid and a non-recombinant plasmid? What's a recombinant plasmid have in it that the non-recombinant doesn't? Lambda DNA. Okay, and remember lambda is where the H gene is, that's part of its DNA. Okay. So how do you do this experiment? How do you go about attacking this experiment? Well, we need to take lambda DNA and insert it into PUC18. What do we have to do to make that happen? We have to chop those DNAs up with restriction enzyme, right? And make sticky ends. So we incubate lambda DNA. How many pieces of DNA do we generate when we chop up lambda DNA with Echo R1? 6 fragments of different sizes, right? When we digest PUC18 with Echo R1, how many pieces do we get? Just one, right? We take that circle and we break it open. Okay? Then if we mix them together and add DNA ligase, what can happen? The sticky ends of the lambda can join with the sticky ends of PUC18 and form recombinant plasmids. But what else can happen? Remember that PUC18 can just rejoin with itself again and form the non-recombinant plasmid? How many different recombinant plasmids can we form? 4, okay? Now that might seem kind of strange because there's 6 fragments on the lambda. But the 2 that were on the ends don't have sticky ends on one of the ends. So there's only 4 fragments that have sticky ends. So we mix them all together. Okay, we mix the digested DNAs from the PUC18 and the digested DNAs from the lambda with DNA ligase, and hopefully we form recombinants. Okay? And then what do? We expose the bacteria, E. coli, to those particular mixes of DNAs, and we do the heat shock and the osmotic shock to try to get them to take up the plasma. Okay? How do we tell if transformation has been successful? Well, the bacteria we start out with are sensitive to ampicillin, right? And do they have the beta-galactosidase gene? Okay. They're LAC-Z negative. Okay. So if they take up a plasmid that is recombinant, how do we know they've taken up that plasmid? They'll grow on ampicillin. What if they take up a non-recombinant plasmid? Will they grow on ampicillin still? Yep, because that PUC18 just rejoined with itself, and it had that ampicillin-resistant gene. So how do we differentiate between the recombinant uptake and the non-recombinant uptake? That's where the blue-white comes into play, okay? Because if that foreign DNA was uptaken by the plasmid, where did it insert? Into the middle of the LAC-Z gene. And it disrupted it. And if that gene is disrupted, beta-galactosidase isn't going to be produced, what color will the colonies be? They'll be white. And if they took up the non-recombinant plasmid, which would just rejoin with itself, the LAC-Z gene is still intact. They'll grow on the ampicillin agar, and they'll also hydrolyze the X-gal and form blue colonies. So what we're looking for after that particular step, which is where you were at the end of last week, are plates that have blue and white colonies. And which color colonies are you interested in for your job? You're interested in the white colonies, right? Okay, so this is a little summary of kind of where you are right now. So in the first 2 weeks of the project, really what you were trying to do is you were trying to make a lambda genomic library. We're trying to make this thing that has all the lambda genes represented in that thing. What is that thing? What is the library? It's a plate. It's a plate with bacteria going on. Which of those plates represent the lambda genomic library?
Which of those plates will contain bacteria transformed with plasmids that have an insert?
Okay. Probably kind of a hint right here that these say control, right? Remember we talked about this last week about what the purpose of these particular controls were? What's the purpose of this particular control? Make sure the E. coli are working. Okay, make sure the E. coli are viable. What was the purpose of this control, which we turned out we needed? Make sure the ampicillin's working. Okay? So we're really down to 2 down here right now. Okay? This is basically a plus DNA plate. These cells were exposed to the plasmid. Okay? And then plated out on LB agar. What kind of cells will grow on there? Will cells that are transformed grow on there? Yeah, because there's no ampicillin in there. But will non-transformed cells grow on that plate? Yeah, because there's no ampicillin selection. Okay? We really only want the transformed cells to deal with. So this is really the important plate for us because it's got the ampicillin selection factor in here. So only transformed cells will grow. And I'll tell you, transformation is not a very efficient process. When you mix those E. coli with those plasmids that you created, very few will take them up. Most of the cells will be non-transformed. So we've knocked all those out by having the ampicillin present in the agar. If you're not transformed, then you're killed by the ampicillin. But what does this part do, this X gap? Now you get the blue and white colonies. Okay? And the blue ones represent non-recombinant colonies transforming the bacteria. The white ones represent recombinants. Now the big question is you've got this genomic, you've got this genomic library, you've got this plate with all these white colonies and blue colonies, you can see them better here. And each one of these white colonies, theoretically at least, has a different piece of the lambda DNA. One of those 4 fragments that had sticky ends. But if I look at this colony right down here, can I tell which piece it has by looking at it? No, there's no little sign that says, hey, I've got the 4878 in here. And you can't tell. All you can tell is it's been transformed with a recombinant plasmid. So this is your library. One of these colonies, a book if you like, has the information you need. But which one? You've got to somehow read these books and see which one has the right information. So what does reading the library entail? What does it involve? Well, you took a white colony. You picked one at random. And if you were doing this for real, what we probably should have had you do is to circle that colony on your plate so you knew which one it was, and probably label that and say, attempt 1. And then you take just a little portion of that colony and do the tests on it, leaving the rest behind. Because if your tests indicate this is a colony that has the right gene, then all you have to do is take the remainder of the colony and grow it up, and your job's done. Okay? You pass that on to the next group, and they've got bacteria that produce the H gene. Okay? So what does reading those particular plates involve? How do you screen the genomic library? Well, you pick a colony. Which color do you pick? You pick the white ones, okay. You grow the cells up in broth so that every time those bacterial cells divide, they form a copy of the plasmid. So all the offspring are clones of one another. You're cloning the gene. Then what you have to do is you have to take those cells from the broth and you have to bust them open and isolate the plasmids. That's what you're going to do this week, and Dr. Morton will talk to you about how you do that at the beginning of lab, right? Okay. But there are ways to isolate the DNA, separate it from the protein, separate the plasmid DNA from the bacterial's genomic DNA. One of the reasons you can do that is because plasmids are small, genomic DNA is bigger. So what technique are you probably going to use? You can centrifuge things, right? Separate things on the basis of different densities. And then once you've isolated those plasmids, you need to analyze them to see which piece of the lambda DNA was in there. Was it 4878 or was it something else? So what methods do you use for plasmid analysis? That's the last week, right?
Yes.
Yeah. Well, one of the things you do is you bust open that plasmid with Echo R1.
Actually, they'll do this at the end of the day today.
They'll do this day today? Okay. And then next week, once you've busted open that plasmid, you'll analyze the DNA fragments that were produced by incubating it with Echo R1. If that plasmid came from a white colony, how many pieces of DNA should be produced when you digest that recombinant plasmid? 2, right? What do they represent? The plasmid, and what's the second part? Whatever was inserted into there, whatever gene was inserted into there. How are you going to know which particular piece of DNA you've got from that digest? You'll run a ladder. And you'll see the relationship between migration in the gel and size. And then you'll look at that piece that represents the lambda gene and say, this corresponds to this spot on the ladder, therefore I know which piece that is. What if you were right? What if you picked the right colony by chance? Great, you're done. What if you didn't? You have to go back to your library and pick out another book. You have to grab another colony, grow it up, digest it, analyze it, and see if you got the right one that time. So it's really a matter of going into that library and randomly picking out books until you find the one that has the correct information. Anything you want to add to?
Yeah, if you were actually doing this in the lab, you wouldn't pick just one colony. You would probably pick 10 or 20 colonies and do
Right.
the growing up in broth, plasmid isolation, and digest. We get around that by having each group in the lab isolate a colony, so the lab group has a greater possibility of at least one in the group having the 4878. Now, obviously, for Monday and Tuesday, that works really well. Wednesday and Thursday have smaller. There's only 4 groups on Wednesday and 3 groups on Thursday, so their probability of picking out a colony with the 4878 insert is lower, but the point is to go through the process of isolating a clone that has this particular plasmid. At one point when I was doing cancer research, I had graduate students and we were trying to clone part of the gene of a calcium channel. And the young man who was doing that had to isolate almost 100 clones before he got the one that had the fragment he was interested in. But that's the way research is. There's a lot of stuff that doesn't work quite right, and you go back and you do it again, or you modify your procedure, all right? But as I mentioned to you, this sort of thing we're doing in lab in these 4 weeks is happening every day in labs all over the world with, slight modifications, fancier plasmids,
Probably a lot of it's becoming automated now.
automation, yeah.
Okay, since we've got a couple minutes, since it's not going to take you all this time to do the quiz, I'd like to follow up on a couple of things. And this is stuff you can just watch, okay? You don't have to take notes on this. This is kind of just additional kind of elaboration. I found another video I'd like to show you on RNAi because it's got some nice animations. A little refresher. Remember that RNAi is a relatively new method that has been discovered whereby cells can regulate their gene expression basically at the level of translation. Because what RNAi does is it allows cells to either selectively inhibit translation of messages or actually chop up the message so it can't be translated. Why did this mechanism evolve? We think it evolved as a defense mechanism against viral infection. Because when cells become infected by virus, the virus come in and take over, and you want to try to prevent that. So RNAi was originally, we believe, a defense mechanism against viral infection that evolved over time to become a method of gene regulation. RNAi, as it turns out, is a great technique that can allow us to do studies where we silence genes and then see what impact that has on the phenotype of the cell. So by knocking out a gene, we can see if the cell changes, then that could indicate what that gene was doing. Okay? So let's take a look at this little animation here. I believe the woman who narrates it is British, and so her accent's a little bit hard to understand a couple of times.
I thought they spoke English.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
So that was really exciting stuff, and has been for about the last 10 years. However, within the last 5 years or so, some new exciting stuff has come out that some of you may have heard of in the news, and that is CRISPR-Cas9. There's a lot of kind of conceptual similarities between this CRISPR-Cas9 system and RNAi. But as it turns out, CRISPR-Cas9 is a much more precise method that you can use to regulate gene expression than the RNAi is. Sometimes the analogy is being made between, like, the difference between a chainsaw and a scalpel. CRISPR-Cas9 can be used to edit genes, not just silence them, and potentially to make genetic substitutions of a very precise nature. And that's really important when people want to consider, can I cure a genetic disease? Some genetic diseases are the result of just a single point mutation, like sickle cell anemia, that alters the function of a particular protein by a single base pair substitution. And this particular CRISPR-Cas9 has the power to make those kind of precise changes. I sent you this morning an article that you can use for optional points, if you choose to, that was in Wired magazine that talks about CRISPR-Cas9. It's not horribly technical, but it does talk about some of the bioethical and philosophical issues associated with that. But then also, there is a short video, if I can find it, that talks about CRISPR-Cas9, and I wanted to show that to you, but Larry, you're good at seeing things on my crowded desktop. Do you see it's a Word document that says CRISPR-Cas9?
Yeah, and it says it has a link to the...
Yeah. Where is it? Oh, there it is. Okay. This one's short.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA, DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So a couple of things that you probably aren't aware of, but are out in the news right now is there's a lot of hope that this particular technology can be used to improve genomes of things like croplands or maybe animals that are used for food. What's interesting is that organisms that have been genetically modified in the past using the kind of recombinant DNA technology that's common right now have been considered to be genetically modified organisms and are therefore under the control of the FDA. FDA has just ruled that because of the way GMOs are defined, that CRISPR-Cas9 technology is not under their purview. And so companies can choose to make genetic modifications using this particular technology, which isn't under FDA control. That's got some people concerned. Point two that you should be aware of. They said that this particular technology has been used in cells from culture, and embryos. And they refer to animal embryos. But it's also been shown to be applicable to human embryos. There's some real concern about that because I think some people could say, well, it would make a lot of sense if we could identify a human embryo that contained the cystic fibrosis gene. And we could then genetically modify that embryo to put a good copy of that gene in there, preventing that particular disease. And everybody go, well, that's probably a good idea. Let's do that. But what if we identify genes for other traits as well, like the gene for being tall? And what if a parent then wanted to have an aspiring MBA child, or WNBA child, and said, I would like this private company to use CRISPR-Cas9 to change and edit the genome of my child. Because they can do that, they can do in vitro fertilization, get the embryo in a test tube, do the technology, and then implant the modified embryo into a parent, and we now have genetic engineering of humans. Yep. So a lot of ethical issues to think about with this particular technology, but this is breaking stuff, and so we wanted to make sure that you've at least heard of it, and if you continue on to the biology curriculum, you'll hear more about it. So hopefully you did a little cheat sheet for yourself of lack and trip off or run. Yeah, they were. I really like them.
Did you just do sort of a general search? Or
Well, I had the one came from the article Sam sent us. The last one did. The other one, I don't remember where I found it.